For decades, peak performance relied on a primitive strategy: swallow a capsule and hope it survives the acid bath of your stomach. We flood our systems with powders and extracts, but the biological reality is inefficient. Many of these compounds break down long before they reach their target tissue, creating a cycle of unpredictable chemical spikes and metabolic crashes. The flaw in this method comes down to an anatomical misunderstanding. We often treat the gut like a simple plumbing system. In reality, it operates as a command center that influences immune tone, regulates hormone signaling, and communicates directly with the brain. Because we treat the gut passively, our interventions constantly fight against our own biology. This outside-in approach has reached its biological limit. Trillions of bacteria, fungi, and microbes live in your digestive tract, competing, communicating, and manufacturing chemical signals that your body listens to every single day. This reality raises a provocative question for modern biology. What happens if we stop trying to feed these bacteria with random probiotics and instead start programming them to do specific jobs? This is internal biomanufacturing. It turns the existing microbiome into an automated biochemical factory, one that produces the exact molecules your body needs right when it needs them. Upgrading the biological instructions of these microbes represents the next major leap in human endurance and cognitive focus. Building this internal factory begins by creating a high-resolution map of an individual's unique biological landscape. This diagram illustrates how that map is constructed. A patient's genetic sequence, their existing microbial populations, and baseline inflammatory markers converge into a single comprehensive profile. This biomanufacturing system relies on exact, personalized data to pinpoint biological bottlenecks, ensuring the intervention solves the right problem rather than relying on generic guesses. Once the map is built, scientists use CRISPR to install new sets of instructions, known as synthetic circuits, directly into the genomes of selected gut bacteria. This flowchart shows a synthetic biological circuit mirroring a computer's if-then logic. When the engineered microbe detects a rising stress marker, it automatically deploys a therapeutic response. These programmed microbes remain dormant until systemic inflammation rises. Then the circuit activates, manufacturing the necessary compounds. Synthetic biology turns these living organisms into automated biological processors that sense and react to the body's changing needs in real time. For cognitive clarity, programmed bacteria synthesize molecules that travel the gut-brain axis, reducing background inflammation that causes brain fog. For physical endurance, engineered microbes can be designed to directly support energy pathways, improving metabolic efficiency during intense exercise, without the need for external stimulants. Instead of the fleeting pulse of a standard supplement, your gut acts as an active partner in the production chain. This results in a state of sustained biological support, a personalized performance engine that manufactures what you need from within. It is crucial to be clear about the reality of this science. Engineering the internal microbiome is a complex medical field, not a consumer shortcut, and certainly not an experiment to try at home. Safety requires strict biological containment protocols, including genetic safety switches that prevent these bacteria from sharing modified DNA with wild microbes, and methods to reverse the intervention if necessary. This technology marks a shift into targeted biological programming, where the effectiveness of an intervention no longer depends on the hope that a swallowed compound survived the digestive process. Peak performance will be achieved by communicating directly with our biology, engineering our internal ecosystem to become an active, intelligent partner in our own health.